welcome to Structural Biology Communications Interviews with Authors. I am John Aguirre from the University of York here in Northern England, and connecting from Universidad de Sao Paulo is Cristy Nonato. Hello, Cristy.
Hello, everybody.
We also have Mark Van Rey from the Spanish Center for Biotechnology, Madrid. Hey, Mark.
Hello, welcome everybody.
Today, we're interviewing the authors of Small Angle X-ray Scattering of Engineered Antigen-Binding Fragments, the case of glycosylation Fab from the Mannitou IgM antibody. First, we got Shubham.
Hi, everyone. My name is Shubham Samuel. I currently work with Dr. Julie Boucard as a structural biologist postdoctoral fellow in CNRS. I recently finished my PhD back in December 2024, and this paper was a part of an obligation as per French regulation for the PhD. So in this paper, I was the one who have written the paper. I have done the SEC-SAXS experiment analysis, and I have done most of the discussions with Julie and Savas on how to proceed with the data collection and data projections in the paper.
Brilliant, thank you. Savas?
Yes, I am Savas Saviris. I'm a professor in structural biology at Ghent University and the VIB in Belgium. And I've worked with Shubham and Julie towards what we have reported in this manuscript in a joint collaboration.
Thank you very much. And to conclude, Julie?
Yes, hello everyone. I'm Julie Boucard. So I am Director of Research at the Centre National de Recherche Scientifique in Lille, the north of France. And so I have been educated as a crystallographer. I'm still a crystallographer. So I'm very pleased to participate to the interview today. Yeah.
Excellent. So we'll start with the questions. And the first one comes from Cristy.
Yes, very nice to meet all of you. So my first question is to Julie. Julie, tell us what sets the Mannitou Fab apart from the other Fabs?
So a Fab is an antigen-binding fragment. That's the part from the antibody that recognizes the antigen. And it sets it apart because it is Mannitou is an antibody is an IgM Mannitou. So they are basically pentamers of what most people know best, IgG like antibodies. So they are, instead of being bivalent, they are decavalent. So they recognize 10 epitopes instead of 2 for IgG antibodies. And yeah, we found that they are understudied, so we were really interested in them. But we also found out on the way why they are understudied. Yeah.
I have a question for Savas. Could you comment on how easy or difficult it was to express it?
The production of mammalian proteins has always been difficult for structural biology purposes. And that has pushed basically the community to go to sources of those proteins that can be closer to the native producing cells of the proteins of interest. Now, as such, the protein of interest in this case was difficult to produce from the get-go. And we basically benefited here to, exploiting as much as possible a mammalian cell-based expression for the purpose of characterizing this protein with the methods we reported in this manuscript, mainly small angle X-ray scattering, which, while does not require tremendous amounts of protein, it still requires I would say, a sizable sample on the order of a couple of milligrams to be able to run experiments correctly.
So when it comes to antigens, non-protein antigens, what makes a good binder to antibodies?
Yeah, thank you for this question, Mark. So here I should introduce indeed Mannitou IgM a little bit more. So it is so IgMs are primitive antibodies and they are natural antibodies, so they are present always. we found that the founders of this antibody, of Mannitou antibody, they found that it binds mannose. So the name is well chosen. Indeed, it's related to mannose binding. And in more recent years, they found it is recognizing highly specifically only mannose in the context of an N-glycan that is called paucimannose. So paucimannose means poor in mannose. So it only contains, in contrast to the high mannose N-glycans, it only contains up to 3 maximum mannoses, so 1, 2, or 3 mannoses. And the most common ones that have been characterized now by glycoproteomics approaches are the N-glycans with 2 mannose and with 3 mannose. And so Mannitou IgM will recognize these specific paucimannosidic glycans with 2 or 3 mannoses. And that's special because these paucimannosidic glycans are not always present. They only appear in inflammation and in cancer, so in tumorigenesis. And usually they are recognized as being malicious, and they should be removed. So that's why you have these IgM antibodies. And so we are also studying that particular characteristic of mannose binding, because we are also, apart from being crystallographer, we are glycobiologists. So we are studying its interactions with Mannitou Fab and IgM.
Very interesting. Thank you.
Well, it's me again. I have a question for you, because in your purification protocol, there are a lot of interesting comments on how you optimize metal affinity chromatography and that you needed to do dialysis. So my question is, what were the challenges that you guys faced during the purification process?
Yeah, so it was clear from early on that the IgM Fabs stand out from the, for example, IgG Fabs in their much lower stability. So they are less stable, whereas a melting temperature of 70 degrees would be normal for an IgG Fab. For an IgM Fab, it's more around 55 degrees Celsius. So so they are already less stable. And this is because they are in a different construction. They are in a much larger protein. And so they need the context of this much larger protein, especially they have an extra constant domain in the heavy chain that is actually stabilizing their structure. Furthermore, we found that in the SEC-SAXS study, so in the paper, we found that we have always 2 species that bind to the metal ion, so the IMAC, the cobalt-affinity purification column. And the second form we also characterize in the paper. So we have the monomeric Fab as we wanted it. And then we could also characterize that in this second peak there was misfolded Fab that we could identify indeed as the Fab, but in a dimeric form. So that the SEC studies were very useful to identify it, so we can remove it from our final products that we will use further for other structural studies.
Thank you, Julie.
Shubham, in the paper, you mentioned the use of GlycoShield as a tool to model the glycan components into the AlphaFold predictions, which do not have modifications, at least the ones done with AlphaFold 2. So how was the quality of the glycan structures assessed?
So first of all, I would like to add more that from AlphaFold 3 onwards, we can add small glycans onto the structure. So in AlphaFold 2, as you mentioned, it was not available during the time we were making these models. But then we stumbled across, well, Julie stumbled across this, that a group, I think it's from INSERM, correct me if I'm wrong, it's in Paris, and they made this software. So what they did was they already did molecular dynamics, extensive molecular dynamics. They saw Ramachandran plots for carbohydrates, and they managed to make 64 constant glycans. So these are the only glycans that you can add. So you can't just make your random glycan. So you are saved from doing all these complex calculations yourself. So rather, what you do is you do mass spec, like we have done mass spec. We know that we have a glycan with like 13 sugars on top of it. We know there are constituencies, like if they are hexosaminases, or if they are hexoses, or like glucoses, glucanacs, what we have in there. And then we launch up the software, we see what is the closest glycan structure that resembles to our native glycosylation pattern. We select it, it runs a solubility assay where it says, okay, in this protein, these are the areas that can be accessed by glycan much more easily, and these are the areas that will be more restricted, for example, for glycosylation to be put on. You give your glycosylation site, it accesses it, it gives you a score on how much occupancy can be possible. And then after that, we put it on the SEC-SAXS using multi-Fox and Fox analysis, as was explained in the paper. And with that, we were able to see that our scores went way better with some models compared to others. So that was the standard protocol that we followed to identify, okay, that these glycan conformations were much more suitable, and well, qualitatively, they were much better and closer to our native structure compared to the others where the score fell off through the roof compared to the ensemble.
We also work a little bit on glycans. When you say mass spec, was this mass spec on the protein plus the glycans, or because we're having a bit of difficulty in getting people to do good mass spec on glycans alone?
Yeah, so in recent years, we are trying to really do glycoproteomics. That means the glycan on the protein. So as usually in proteomics, when you do bottom-up proteomics on your purified protein, you will chop it up using trypsin, and then you have peptides and you have glycopeptides. And so these all go into the liquid chromatography, and then from there go into the mass spectrometer. So then you can analyze not only the structure of the glycan, or at least the sequence, and but you can also see on which site it is glycosylating. So that works actually really nicely. We have now collaboration, and so we want to do that like routinely, and I think that would be really nice to continue doing that in the coming 10 years, let's say, to have always purity of protein, to have an extra quality check using mass spectrometry, especially when we have glycoproteins in the epinorth, that we know also how they are glycosylated. They will not be homogeneous. They will not be fully homogeneous. But I'm not sure that whether you asked about the glycans.
Well, I was just wondering if there are groups that are especially good in doing mass spec of glycans. In our case, they are, well, for instance, lipopolysaccharides and teichoic acids that get hydrolyzed, and we would like to see what the reaction is. So that's maybe a little bit different from what you do.
Yeah, I think that's so. What I do is actually not what most people do here in Lille. So most people do what you are asking for. They use MALDI, different types of MALDI, to like Kitov and other ones. So more on lipopolysaccharides, or they also have FTICR MS. Where you can work with the glycolipids if you need to. So no, they originally we chopped off the glycans using PNGSF or beta elimination for the glycans, and then did permethylation and MALDI-TOF. So that's the real expertise of Lil. If you
Okay, I will look into that. Yes, I
come
will
to
definitely
Lil,
look into
yes.
that.
Yeah.
That was an important part I thought about, where you distinguished between the kissing fabs and because you had like a dimeric, well, contaminant is not the right word. You had like a fraction that was dimeric. And you ruled out on the basis of SAXS and maybe some other experiments that I didn't understand completely, that they were not the kissing Fabs interacting with each other partly through the glycans, but that there was some kind of strand swapping between the Fabs. I was wondering if you could explain that a little bit more, how you could really rule out one and not the other.
Like how kissing fabs work is, so normally whenever we throw two proteins together to form like a complex or into the alpha fold, what it does is that based on evolutionary models, it tries to figure out which are the most reactive surface or which would interact by default, and then try to make interaction out of it. So for Fab, being its active domain, which is the most important part of a Fab and the part that interacts with the antigen. So what it was always doing is it was putting those two antigen parts together with the body apart. So it looked like the Fab was trying to come face down towards itself with its body being to the back. And we kept getting it on a repeat every time we tried, no matter if we tried AlphaFold 2 or AlphaFold 3 or even like collab fold and other things. So I started, out of frustration, I started calling them kissing Fabs because we were in Paris. So I said, okay, we were lucky. So I started calling them kissing ones because they kept coming together to interact. But then we looked into literature, we looked into crystallographic structures, and we couldn't find anywhere in the literature where Fab would come like this and try to cross interact with each other. And then we figured out that the glycosylation site is actually at the bottom. So if you look at the structure, the glycan comes out from the bottom, not from the top. So that makes, so normally then they should interact like this if they are interacting with each other rather than interacting face like on a head-on collision with each other. And then we also did a CD, circular dichroism experiment, which is also in the paper towards the very end. And there we saw that when we heated the protein, we got a similar absorption pattern compared to what we got for the second peak. So from there, we delineated that maybe it's more like a aggregated form, or it's more like a dimer swapped version where they are completely swapped to each other rather than being two distinct species holding their own identity.
Yeah, and maybe to add to this, I mean, the domain swapping scenario is not just thought as by accident. There are examples in the protein structural databases of domain swapped Fab fragments. So it is, let's say possibly a folding artifact that seems to be supported by certain Fabs. And so, in trying to explain the data that Shubham was collecting, this seemed to be one of the possibilities. Again, it remains a hypothesis. It's not fully corroborated at this moment.
So how does the glycosylation pattern differ from other IgM-derived Fabs? And is that, if there is a difference, is that significant for the function?
There is very little known about this. So about glycosylation of IgM molecules in general. Interestingly, there has been a paper last year in Nature on the glycosylation of IgM in COVID patients, so that are exposed to SARS-CoV-2. And they find that the glycosylation there, those patients that have a high severity of the disease, they have more hymanose and sialylation in their glycans. And this correlates with the severity of the disease. Also, the glycosylation can differ depending on when the antibody is expressed in what context. But in our study, we find a complex glycan on the Fab. So it is conserved, this N-glycosylation, in in the constant domain 1, the first constant domain of the IgM, and we don't know its function. So we only observed that when we Shubham did the multifox analysis of to fit which glycan conformation was best fitting the SAXS molecular envelope, we found that if we don't add the glycan using glycoshield to the Fab, then it would happen that in the multi-state conformers, that in one of the conformers, the light chain and the heavy chain would dissociate. So, and this can be totally artificial, of course, but it also indicates that probably without the glycosylation, the Fab, IgM Fab is less stable again. And yeah, it's probably has the function at least to stabilize in the interdomain interface the Fab molecule.
Thank you.
Now it's my turn with a more general question to Savas. What could be the implications of this research for the future of immunological research?
Thank you, Christy, for this question. So I think, as already very well mentioned by Julie and also alluded to by Shubham, the scientific literature and knowledge on IgMs in general is quite poor relative to other types of antibodies. So I think as such, if one provides a biochemical and structural context on at least part of an IgM, in this case, the Fab for Mannitou, that's already a major contribution, I think, for people to lower the threshold for studying these kinds of molecules. Now, I think what is especially fascinating about this particular IgM Fab is also its glycosylation state. I should note we have worked in my laboratory on many other Fab fragments that are typically not glycosylated. So the fact that you have one in this case that is glycosylated, I think is already important information for the field, and also in the way it has been characterized nicely in this work will probably help others to view their work in a different way. But let's say moving forward, I would say what has now been achieved by this work, the ability to produce this protein to some reasonable amounts that are stable protein, also opens the avenues to investigate it with other structural methods, not only the ones that we have highlighted in the manuscript, but also now to push forward with X-ray crystallography and get a more accurate and detailed structure to atomic resolution as much as possible, including possibly delineating the way this Fab interacts with its carbohydrate antigen, which I think is going to be very exciting. So we've learned quite a bit, but it also paves, this work paves the way for future work, which is actually currently ongoing in our groups.
Fantastic, Savas. Thank you very much.
Right. Next question to Julie. This Acta Cryst publication is a paper on a small angle X-ray scattering, so SEC-SAXS, of macromolecules. How do you see the future role of SAXS studies in structural biology and crystallography? But also, how can SAXS contribute to the sample quality in cryo-electron microscopy?
So that's something I have been thinking about, because many of the proteins, as Savas was explaining, are now produced in HEK cells, HEK293 cells, because that's where they fold correctly. They often have the correct glycosylation and so on. But these expression systems, they might take a couple of days. So after transfecting the cells, you need 3 to 4 days to have a good expression. And so you have byproducts, you have misfolded protein there that when maybe it was produced the first day, and by day 4 it's already degrading or breaking down. So you need to be able to look at the heterogeneity of your sample, and this is very important. In crystallography, usually if you have a heterogeneity, it might still crystallize out one of the forms, and this misfolded part may be completely ignored because you need to fit one conformer in a crystal lattice. But in cryo-EM, you will see that this heterogeneity, if you just go for it, can be large. So you want absolutely to avoid to have misfolded proteins or other oligomer forms like dimer or Fab in your preparations before you go to the cryo-EM. So I think that SAXS can be more and more can be ideally used in conjunction with other structural studies like on the cryo-electron microscopy.
Brilliant. Thank you, Julie. Sabas, so for us, this is the first interview with researchers from different laboratories and about a project that was an international collaboration, which we are very interested in. So how did the collaboration come about and which parts of the project did each of the labs undertake?
Actually, this came about in the context of a joint PhD between our respective universities and institutions, which united funding from both institutions, but including co-funding from the European Commission through the Pearl program, which was hosted by the University of Lille. So I would say in a first instance, it was really a convergence of resources and the format for hosting a PhD jointly in our laboratories. And then, of course, identifying an appropriate international candidate. In this case, it was Shubham who was selected. That allowed us to basically unite our forces. But I would say, let's say from the scientific point of view, this was, I would say, more like an inevitable evolution because, so Julie being a recognized carbohydrate glycobiologist researcher and our laboratory being more recognized in a more general structural biology context of, especially in the context of antibody-based structural biology in complex with antigens and having the technologies to express such proteins kind of brought us together to host this particular research. So I would say it was organically grown, but also directed by the funding resources that we had available to make this possible. And of course, finding appropriate people to work on such a project, in this case, Shubham Semwal. So that's it, I guess.
Thank you.
So related to that, I see that Shubham is affiliated to both labs.
Mm-hmm.
Did that bring any specific challenges or advantages?
Yes, of course. When you have multiple hosts, multiple situations, that brings challenges, but that also brings as well a richness into the process. So I would say, let's say from a supervisor's point of view, the challenge had been to train a PhD student and coordinating on the research activities that were not 100% happening in our laboratory. And of course, seeing each other only part of the time and coordinating for the rest of the time online and via email. But then I would say there has been a clear added value to this, hosting a PhD student who is, jointly guided by another colleague, and also bringing in that knowledge into our group and sharing our knowledge in the reverse direction. But maybe I should direct it now to Shubham. Maybe he has some views on this because he has experienced this for the last 4 years.
Yeah, so I completely agree with Sebas that it was more on the positive end that I could experience working in both in France and Belgium, and in two very big laboratories that have their own mix. So for me, it's almost like a music composition where you have different people coming around and making a symphony together. So it doesn't matter who is the guitarist or who is the lead singer, but if they play their role perfectly, it comes as a nice harmony. But it does come with its own challenges also, and mostly, in my opinion, it was in terms of how both labs see their work values. Because, for example, in France, we are limited by the time. It's a 3-year PhD project, so everything is supposed to be rushed, and so people have a different work attitude towards working. They are more like, okay, let's work. So less time thinking about the details and the minute things, it's in bits, and more about doing things and then getting to understand why it works and how it works. Whereas Belgium, people are much more meticulous, like they have time, so they really go in depth. They talk out about everything in much more detail because they have more resources and more time. Specifically in SEC-SAXS team, they have much more thing to go. So they were both awesome things in their own respect, but yeah, to manage two things coming apart. So like there was one fun incident that the first time I presented in SEC-SAXS group, there were so many questions asked, like it was almost like what's going on, because they were like, have you read this research? How can you do this? Have you got into depth of this and that and that and that and that? And I've been presenting a little before. And yeah, in France, nobody asked me to go in that much depth. So then later I started to read research and reviews into more depth. So it was a bit more taxing, but yeah, it was fun also in the end. So yeah, the two cultures, yeah, it's like two different cultures coming together of science. So that was a bit that took time to taking used to, but apart from that, it was a very fantastic approach to go to a dual PhD program.
Great, thank you. That was very interesting.
Yes, we actually wrote a editorial on supporting international collaboration. So this is great to see this kind of work.
Yeah.
So my question is to you, Shubhan. Learning more about your personal experience, what keeps you motivated in your research, especially during challenging times?
So what keeps me motivated? It's a really good question. So I actually have a very innate love for biology and for biological workings. That's what pushed me from leaving my engineering diploma and entering into the world of PhD and going into hardcore science. And times have been tough, but I like to discuss, I like to talk a lot. So whenever I get pushed down, I get depressed, I always approach my supervisors and I'm like, okay, what is, give me just one thing that I can hang to my dear life and move forward on it. And mostly Julie and Sevas have something good coming out of it. And then they say, but out of this, what about if you learn this? And then like when I'm at the lowest, I just cling to it, like, okay, this is the only one point that will take me somewhere. And mostly it does. And that's what keeps me going, I think, is just the love. I know it's a very cliche answer, but yeah, I was groomed. Yeah, because my mom is also a professor in biology, so I was groomed from a young age to be curious about biology, and that's what keeps me going more and more towards it.
Thank you. That's interesting. Actually, you also already told us an interesting anecdote. Do you have any an additional one you would like to share?
So there was one time when, so because we were trying crystallography also in parallel to SAXS data, and with crystallography I learned, because I was learning crystallography as I was doing it. So like normally when you see papers, you see beautiful crystals and you are like, okay, I will get one of these crystals and I will go and fish it and we will have a nice data set. And for me, my first time was disastrous because we went to Soleil. It was full of ice. I couldn't fish a single thing. I was like, okay, when is our train to back home? And then later, like after 3 years or so, I fished a crystal. It was looking super dirty, super like, it's not the prettiest child you can imagine to have in the mix. And we fished it because we said, okay, we have nothing more to do and we have a beam time coming. So we fish and we send it to Petra. And it diffracted actually. And I was surprised. I said, that's because we thought it was salt crystal. We always have that condition with salt coming up again and again and again. And that one time it was a jackpot. So now we are trying to optimize it, like Seva said before, also to get more in depth of this Mannitou to study. But this was a hard learned lesson that in crystallography, you never judge a book by its cover. Like even the dirtiest looking crystal might give you the data you are looking for so hard.
Yes, yes. And unfortunately, also the other way around.
Yeah.
Sometimes you get really nice crystals that diffract only to very low resolution.
Yeah.
I just wanted to ask Shubham, how did the Viva go, the PhD exam?
Actually, surprisingly for me, it was really nice. I enjoyed it thoroughly. I mean, I was really nervous before, but because I defended in both universities, I defended in Belgium and I defended in France also. So when I defended in Belgium, because it was an internal interview for like 3 hours, if I'm not wrong. Yeah, it was around 3 hours of questioning. I was like almost nervous, but the jury was super friendly, and it was more like they were guiding me like towards the correct path rather than bashing me around for what I did and what I didn't. So I found it actually pretty enlightening and pretty like, yeah, it was like, normally I've heard people saying, oh, PhD, they completely kill you, they butcher you around and you feel humiliated. But no, I actually felt more at peace and like more connected to what I was doing than before. So I thoroughly enjoyed it. And then when it came to my public one, I was completely ready when it came to France. So at that time, I knew I could own it, because if you have gone through 3 hours of questioning, there's not much left to to go
Yeah.
around to that point.
Well, I think it's my turn, and I actually I'm gonna ask to you all because we are human beings. So I want to learn from you guys. Do you have any hobbies, activities that help you maintain balance and enhance your work in research?
So I started off with an association for nature, so for protection of natural places, and see that there's diversity, biodiversity in these places, etc. And then at some point, I said, okay, we are not getting anywhere. So I also went into politics to try to have more attention for these parts, especially on the local level, because I believe you have to start this on the local level and then go slowly to the national level. So some of my colleagues have progressed to that, but I purely keep that as a hobby. So it's really a hobby for me. So I contribute, we discuss, I contribute as a scientist, maybe as a biologist, and that's what I try to do. But it remains a hobby for me.
Maybe I can say a few words. So obviously, I'm a very busy man, but at the same time, I find refuge to cooking for family and friends. I'm the designated cook at home, but I do see it as a relaxation moment. It's also existential. You have to make something to to eat, but at the same time, I put some effort in it, and I really enjoy fusing the different, let's say, cuisines that we are exposed to, and also love to have friends and family over for elaborate dinners and lunches and barbecues. So I really like to go into these kinds of activities. But let's say the other side of what I do is in of the very little free time I have, I actually play basketball. I've played basketball for 4 decades now, believe it or not. And I keep playing, of course, at what I can do with a relatively young group, and that keeps me a little bit younger than I look at the moment. So that's it.
I should share with you some Brazilian recipes so you can try that.
Sure, that would be nice. Yes, it's an area of culinary space we haven't visited yet.
Shubham.
As for me, I have a fascination towards poetry. So I do write in Urdu and Hindi, so two of my native languages. But I write something called shairi, so which is like a short stanza of poetry, which are not lyrical per se, but they depict the emotion of life. So they can be happy, they can be sad, they can be romantic about it, and they are normally like a stanza or so. So they are like 6 to 8 liners mostly. So I do that, and when I'm feeling lazy to put my brain into it, I like to play video games a lot. So I have a collection of PlayStations at my home with lots of games. So I'm an avid gamer. So mostly like I have a routine of playing like at least 1 hour in a normal free day to get my brain completely rebooted for the next day.
Okay, what's your favorite video game then?
Elder Ring.
Okay, I must say that was a very pleasant conversation. Thank you very much, everyone, and best of luck for the future.